Thank you very much. Dear colleagues, allowed me to present the results of our group, which the title is a pro, pre, and symbiotic supplementation in oxalate homeostasis with a focus on microbiota and oxalate degradation activity. Okay. I'm sorry, my screen doesn't move.
We're waiting, it's okay.
Oh, okay, it's okay. It's okay right now. Urachiliasis is a common condition that affects 1 out of 11 people in US and around the world, with the recurrency rate about 50% within the 5 years after the first onset. And here on this map, you can see this so-called stone belt. You can see this in a red color, and it presents the numbers of up to 10 to 15% of the people, of the patient with their kidney disease all around the world. And we observe increase the number of people with the kidney disease, and the main reason is habits, dietary habits, climate, environment, ethnicity, and heredity. And among the most significant are poor oral fluid intake, high animal-derived protein, high oxalate intake, and we like to drink tea, like soda, eat chocolate, coffee, and all of this food contains a high level of oxalate, and of course high salt intake in our diet. All of these increase the possibility to get their concrement. And here you can see the different size and shapes of the kidney stones, and their size can be from micrometers to several centimeters. And mostly in 97% they locates in the kidney, and among them all of them can be composed from different minerals, and worth to mention that calcium oxalate has a prevalence up to 80% among other biominerals. Because of that, their oxalate in hyperoxaluria was the main topic of our study, because of the frequency of these chemicals. And worth to mention also that even we know that naturally the small concrements can pass out with the urine from the body, or in more difficult cases it can be out by the surgical removal. But despite of that, the stone does not, the removal of stone does not eliminate the cause of the disease. And many patients suffer recurrence that push us to think about the type of conservative treatment and preventive therapy. As I mentioned, that calcium oxalate is one of the main minerals which compose the kidney stone. Oxalate per se is a potentially toxic anion of decarboxylic acid, and which is widespread in both in plant mammals. And in our body, of course, in normal condition, we can keep the oxalate homeostasis, and this homeostasis provided mostly by their kidney up to 90%, and very important, and it was actually our main topic, their gut can also participate in the oxalate homeostasis up to 10%, and excrete with their feces their oxalate. High oxalate diet and gut microbiota violation can result in hyperoxaluria. Then we observe the increase their oxalate concentration in the mostly in the urine up to 44 milligram per day, and calcium oxalate stone formation. In turn, kidney function decline in non-stone formers leads to a decrease in renal oxalate clearance and increase in plasma oxalic acid concentration, according to the progression of chronic kidney disease stage. Despite, as you can see here, that intestine is responsible for only 10%, but then we look more precisely in the metabolism of oxalate in our organism. We will be surprised, but intestine plays a very complex role in this homeostasis. Since in our intestine we have the several intestinal epithelial oxalate transporters, which are responsible for both secretion and the absorption of oxalate. And also, which is very, very important, all of us very familiar with this topic, the significance of microbiota. And intraluminary in the gut, the microbiota plays the essential role in the metabolism of oxalate. And despite the potential toxicity of the oxalate from the body, it's interesting that humans lack the oxalate metabolizing enzymes and are not able to degrade excessive oxalate levels. And in this case, it's obvious if we have the problem with our microbiome, we can have the problem with the metabolism of oxalate. And microbes, they contain the two main oxalate degrading enzymes, and as you can see here, humans intestinal oxalate degrading bacteria complement the missing oxalate metabolizing ability in a mammalian host, are collectively identified as a oxalate metabolizing bacterial species. And this group divided in two: narrow profile oxalotroph, and nowadays only one bacteria belongs to this group. Oxalobacter formigenus is the only specialist oxalate degrader in the humans, which use the oxalate as the sole source of carbon and energy. And also we have the longer list of microbes, which can be titled as a broad profile oxalotroph. And among them, we can see Lactobacillus bifidobacterium and so on. And they also contain the genes which can degrade oxalate. And these two groups, they are the main topics and main interest of our study. As you can see here, I've just took from the PubMed information about the number of publications for the last almost 20 years, for the number of publications using their keywords probiotic and urine stones, and not too many actually publications, despite their possible significance of probiotics or symbiotics to prevent the kidney stone disease. And the main idea of our study was to test the hypothesis that the total ability of intestinal oxalate-degrading bacteria is very important in comparison to the number of oxalate-degrading bacteria, since you saw on my previous slide that we have the numbers of bacteria out of just Oxalobacter formigenes which can degrade oxalate. And the question which we raise is the relationship between the consumption of oxalate rich food and the number of ODB. How does oxalate affect the mammalian gut microbiota and in general, and how does impaired gut microbiota influence the overall oxalate metabolism of the host, since the majority of previous research have focused on the role of individual taxa in oxalate degradation. Okay, taking into account the fact that a lot of people talk about the significance of diet and the kidney stones disease, in our first part of experiment, we check how their diet, high level of oxalate diet, which we induce by gavage the rats with a 2 milligram per kilogram sodium oxalate for 14 days, how it will affect the number of oxalate degrading bacteria. And we use a very simple experiment, which I like, the simple experiment, which is easy to reproduce. We took just two different media, one media with their only source of energy as an oxalate, and we use the concentration of 5 grams per liter. And also we use the widely used medium, and we add to them also the oxalate, and we compared the growth of the fecal bacteria from the rats on this medium. And as you can see on this slide, the A figure represents that the number of oxalotroph specialists, this is the Oxalobacter formigenes, and also their facultative oxalotrophs. And we can see then we use the high oxalate diet. It's clear that number of Oxalobacter formigenes increase and the number of lactobacillus bifidobacteriums decrease because they don't have enough source of energy.